Good morning, everyone, and welcome to this seminar arranged by the Russian-Finnish Life Science Center. And it's a very nice weather here in Turku, where I'm speaking. My name is Kalervo Väänänen. I'm chairing and moderating this session. And our topic today is application of genetic testing in medicine. We will hear 5 excellent speakers, and we have about 10-12 minutes presentations, and after that questions if you would like to present. And our speakers today are Juhani Junttila. Juhani, say hello to. We have also Artem Lada.
Hi.
Katri Selander. Julia Gudrichova and
Hello.
Julia Sopova.
I had my pre.
But before we go to the presentations, scientific presentations, Yelena Petrova, who is actually chairing this Russian-Finnish Life Science Park, will very shortly describe our project. Yelena, please.
Good morning. Good morning, dear colleagues. I am pleased to welcome you to today's online scientific conference on molecular genetics in medicine. Thanks to digital resources, we have guests despite quarantine activities. The conference is organized by the municipality of Kotka, a partner of the project Russian-Finnish Life Science Park, as part of the cross-border cooperation program South-East Finland, Russia, 2014-2020, together with the lead partners, St. Petersburg Technopark. The goal of the Russian-Finnish Life Science Park is to develop scientific and cross-border cooperation between Finland and Russia, to put the cross-border region as a development area of life science, and also to strengthen the internationalization of small and medium-sized enterprises by stimulating technology transfer and cooperation in R&D, as well as leading collaboration between academia and authorities. Application of methods of human genetics helps to uncover the cause of many diseases, allows to create new effective diagnostics methods, and by studying the genome of a specific individual to create a personalized approach to his treatment or pharmacological correction. Molecular genetics methods underlie the creation of modern recombinant pharmaceuticals, vaccines, monoclonal antibodies, cell cultures producing biologically active molecules. This segment of the pharmaceutical market is currently developing rapidly, and here the most significant events are taking place. For the effective treatment of many diseases, primarily of the immune nature, point effects are required, sometimes at the level of individual cells. The creation of target-oriented drugs, including conjugated and DNA vaccines, will increase the effectiveness of the treatment of cancer, rheumatism, and infectious disease, including viral ones. As life itself has shown, in the context of the coronavirus epidemic, all over the world there is a need to create and produce effective diagnostics test systems for virus detecting. These systems are also based on molecular genetics. Therefore, the theme of our conference is very relevant. We are going to discuss current issues in the field of medical genetics. Leading experts in the field of medical genetics were invited to participate as speakers at our conference. We also invited specialists from manufacturing enterprises to discuss the most important issues in this area. Let me declare our conference open. Now I give the floor to the moderator of the conference, Professor Kaler Vavanen. Please.
Thank you very much, Jelena. And I think that we will go now direct to presentations. First speaks Professor Juhani Junttila, and he's Professor of Cardiology in the University of Oulu, and well in this area. And he will speak genetic background of myocardial fibrosis related to sudden cardiac death. Juhani, please.
Thank you, Kalervo, and thank you for the invitation to speak in this conference. This is an honor. Can I have my slide? So my topic is the genetic mutation related to myofibrosis and sudden cardiac death. Next slide, please. And here are my disclosures. Next slide, please. Cardiovascular death is the most common cause of death in developed countries in the world, and approximately 50% of all cardiovascular deaths are sudden. 70% of cardiac death are caused by coronary disease, but the proportion of coronary disease as a cause of sudden cardiac death has been declining during the last 20 years, as you can see in the figure on the left. In the meantime, non-ischemic causes or cardiomyopathy causes of sudden cardiac death, the proportion has been increasing. The overall incidence of unexpected sudden cardiac death has remained stable. So even though a lot of emphasis has been put on treatment and diagnosis of coronary disease and cardiovascular diseases, it hasn't translated into decreased number of sudden cardiac death. Myocardial fibrosis is a common feature in almost all cardiovascular diseases, in coronary disease, and also in non-ischemic causes of sudden cardiac death. Next slide, please. Here you can see a figure of an autopsied heart, and the white parts are fibrosis. As I said before, fibrosis is a common feature in pretty much all cardiac diseases, which is mostly evident in cardiomyopathies, and the common causes of sudden cardiac death due to cardiomyopathies are dilated cardiomyopathy, hypertrophic cardiomyopathy, and arrhythmogenic ventricular cardiomyopathy. And fibrosis is prominent in all of these cardiomyopathies. Next slide, please. What got us interested on the fibrosis causing sudden cardiac death was this family. In 2004, a 14-year-old girl died suddenly in downtown Oulu, and in the autopsy, a myocardial scar in the inferior posterior wall of the left ventricle was detected. Her mother, a couple of years earlier, were resuscitated from sudden cardiac arrest, and also she had a scar in the inferior posterior wall of the left ventricle in the cardiac MRI after the sudden cardiac arrest. Next slide, please. Here you can see the histopathological findings of the 14-year-old girl at the autopsy. On the left side, you can see the scar area. The pink area is fibrosis, and the red area is myocardium. And you can see the difference between the amount of fibrosis of an age-matched accident victim on the right side. So there is a considerable amount of fibrosis in the left ventricle. Next slide, please. In this family, we found a lamin AC gene mutation, which causes the myocardial scar in the young girl and the mother. And it was a de novo mutation in the grandparents. Even after paternity testing, we couldn't find the same mutation. So the mother had the mutation as de novo, and the daughter had it, and she died before having kids. So the mutation actually died with her. Next slide, please. So we got interested on how prevalent this kind of cause for sudden cardiac death would be, and we went through our study population, which is the Finnish genetic study of arrhythmic events called Finchester. We've been collecting sudden cardiac death victims in autopsies since 1998, and around up to 2017, we have over almost 6,000, 5,869 autopsied sudden cardiac death victims, which we have collected clinical data and ECGs prior to death from about 1,000 of them, and DNA and RNA from formalin-fixed paraffin-embedded tissue samples. Next slide, please. What made this possible is the Finnish law of investigating a cause of death. In Finland, it's mandatory to do an autopsy, a medical legal autopsy, if a death is sudden and unexpected. And you can see here in the figures that the amount of sudden deaths autopsied in Finland is much higher than in a lot of the Western countries. Next slide, please. The definition for this kind of myocardial fibrosis is presented here. We came up with this definition according to the autopsy findings of the 14-year-old girl, and we used them the same in our prevalence study. So no signs of coronary artery disease in autopsy or valvular heart disease, no signs of other inherited structural disorders, all these cardiomyopathies, hypertrophic cardiomyopathy, dilated cardiomyopathy, and ARVC, no myocarditis, and no hypertrophy at autopsy, and no other disease explaining the fibrosis. Next slide, please. What we found in this population is that in the young subgroup of sudden cardiac death victims under 40 years of age, fibrotic cardiomyopathy with this definition that I showed you earlier was the most common cause of sudden cardiac death in the young, with 28% prevalence. Next slide. And this finding has been shown in other populations in the world also. Here's a study from UK where they found in athletes who died suddenly is a 16% prevalence of left ventricle fibrosis without any common cause for the fibrosis. Next slide. In the total population of the Finchester study, primary myocardial fibrosis was the cause of death back in 2012 in 145 subjects. And like I showed you before, it was the most common cause in young sudden cardiac death victims. Next slide, please. We collected the best quality DNA from 96 sudden cardiac death victims with the cause of death as primary myocardial fibrosis. And we performed next generation sequencing with an Illumina NextSeq 550 platform panel of 174 myocardial structure and ion channel genes. And all the detected variants were classified as pathogenic, likely pathogenic, or variants of uncertain significance according to the American College of Molecular Genetic guidelines. And we took out all variants that were over 1% minor allele frequency in the results. Next slide. About 10% of the sudden cardiac death victims with primary myocardial fibrosis had pathogenic or likely pathogenic variants. And all of these variants have been previously associated in literature with hypertrophic cardiomyopathy, dilated cardiomyopathy, or arrhythmogenic ventricular cardiomyopathy. But these sudden cardiac death victims didn't have any of these findings, classical findings of these diseases in autopsy. And there were no ion channel mutations that we found in this population. So this suggests that these variants cause primarily fibrosis, which is expressed in all of these before mentioned cardiomyopathies, and it's enriched in sudden cardiac death population and sudden cardiac death families in our population. Next slide. Here's a table of the findings, and you can see in the red, these variants have been previously associated with hypertrophic cardiomyopathy, in the yellow with dilated cardiomyopathy, and with green with arrhythmogenic ventricular cardiomyopathy. And you can see the criteria and the score we used for determination of the pathogenicity of the variant. Next slide, please. Here's a list of the variants of uncertain significance. There are a couple of these which have not been published before, and this is something that we're looking into more in our future studies. Next slide. Here's a histopathological example of the autopsy findings. On the left side, you see myocardium with stained pro-collagen type 1 antibodies. So the brown area is fibrosis, and you can see the clear distinction of how much fibrosis in these cases you can see in the histopathological findings compared to an age-matched suicide victim. So the amount of fibrosis is extensive. Next slide. So as a conclusion, ladies and gentlemen, many victims of sudden cardiac death due to primary myocardial fibrosis have mutations in myocardial structure encoding genes, and studies in first-degree family members who are living is of major importance to find out subjects at risk and whether we could prevent sudden cardiac death in these families. It seems that primary myocardial fibrosis and all these classical cardiomyopathies represent a different manifestation of a disease spectrum in which genetic predisposition of the disease and myocardial fibrosis are the common core of the disease development. And what we're doing now is we're going through first-degree family members of these pathogenic and likely pathogenic variant carriers, sudden cardiac death victims, and we've been doing echocardiograms and cardiac MRIs to detect fibrosis in these family members to prevent sudden cardiac death in the future. So thank you. I'm ready for questions, Kalervo. So let's go through them.
Thank you very much, Juhani. And maybe I ask the first question that if you think the whole sudden cardiac death, how many percent of these fibrosis cases represent from the whole sudden cardiac death cases?
At the moment, I think that in sudden cardiac death victims as a whole, these it's about 3 to 5% in different populations. So it's a small number, but what we hope to find from these patients is the knowledge of how fibrosis is accumulated in all these type of cardiac diseases, and how we could prevent and detect fibrosis in the population, so we could prevent sudden cardiac death also in coronary disease patients. Because in coronary disease patients, fibrosis is also a major risk factor for sudden cardiac death. So it's in 93% of all sudden cardiac death victims, we can find fibrosis in. So fibrosis is a common feature in all sudden cardiac death victims, which we need to find out and prevent.
Do you think that obviously there are several gene defects that are behind that fibrosis? Do you think that we know most of common already, or are we still looking for?
I think we're just in the beginning to find them and understanding on what the variants actually do. Because in the exome, if you would, that's something that we're doing also in the future and right now. Then if you do an exome sequencing of these sudden cardiac death victims, you always find about 16,000 variants that might be causative. So it's a wealth of information that we're not really sure right now what causes what, but we need these family member studies to kind of find out the pathogenicity of a lot of these variants. And that's something that we're lacking behind in a lot of the genetic studies at the moment, in my opinion.
How do you see the future? Do you think that there will be, at some point, we will screen this type of diseases, although they are quite rare, but affecting young people at the population level?
Yeah, we're pretty far from that at the moment, and it's a complicated question. There's a lot of issues on screening at the moment in the world regarding athletes, like ECG screening or echocardiogram screening, and what really needs to be determined is that we have to have something first that we can prevent, which we can prevent the sudden cardiac death from happening before we start screening. And at the moment, it's only an ICD pacemaker, and that's something that we cannot put to everyone. So we need treatment, new treatments also for this before we can start screening general populations.
I see. Okay, I think that I see any further questions from. And at this point, I thank you very much, Juhani. It was a very interesting and extremely important topic. And then we go further. Now we will start focus more like one gene, but it's the big one. And Artem Lada from University of California will next presentation. Artem, please.
And also for all the organizers who invited me to talk today on this wonderful meeting. And I do apologize in advance if I say something stupid. It's because it's about midnight here in California. So my brain doesn't work absolutely 100% efficiency. But anyway, I'll try to do my best, and we'll talk about the BRCA2 protein. So just to give you a little bit of introduction about the topic. So I work in the laboratory that is focused on the studies of molecular mechanisms of DNA recombination. And this is a picture which all of us saw as early high school, I believe. This is the first model of homologous recombination being produced already in 16. And so it basically states that there are two chromosomes, they can cross over, result in exchange of exchange of genetic material. So now, in the past 100 years, this model been extensively developed, and now this looks like this. This might look a little overwhelming at the first glance, and because of this, I'm going to focus on this part, which I'm going to zoom in a little bit. So this is the first stages of homologous regulation, which is a focus of my research. So now, if you have a double-strand break in the DNA, one way to repair it by means of recombination. And in this process, first the ends of the break are getting resected, which results in the formation in the single-stranded DNA ends. And then these ends can invade into the homologous duplex DNA, shown here. So now there is a plethora of proteins which are involved in this molecular processing of double-strand break. For the sake of this specific talk, I'd like to focus on two proteins. And the first protein, called RPA, shown here, it is a single-stranded DNA binding protein that will cover this single-stranded DNA very quickly. However, on the next step of the molecular process, this RPA has to be substituted for another protein, which is called RAD51. Now, RAD51 is actually a molecular recombinase. This means that it can mediate the strand invasion here into the homologous duplex DNA. Now, here there is one problem, that the complex of DNA in blue here with RPA, structurally you can see it here, is very tight. So in order to replace the RPA shown here on left for RAD51 protein shown here on right, you have to somehow kick out RPA from the DNA, and that's not easy. So it's been shown in our laboratory previously that the topic of today's talk, BRCA2 protein, can actually help in this protein exchange. So BRCA2 is able to bind to RAD51, shown in blue here, and then by using some magic, it deposits it on single-stranded DNA, thus replacing RPA for RAD51. All right, so now I'm going to switch gears a little bit and talk about what does BRCA2 has to do with actual molecular medicine. So as we all know, this designation stands for breast cancer susceptibility, and the protein is called so because the corresponding gene has been identified in families with high incidence of breast cancer. If you have this mutation in BRCA2 gene, your chances of developing breast cancer are significantly higher. And on top of that, in actual tumors from patients, very often people find somatic mutations in this gene. Now, it's also important to point out that BRCA2 is a classical example of molecular double-edged sword. Because from the one hand, if BRCA2 protein is mutated, it can drive tumorigenesis. However, on the other hand, once the tumor already formed, if it has certain mutations in BRCA2, they can render this tumor susceptible to certain lines of therapies, and thus can help in treatment of those patients. But in any case, it's important to know what mutations are found in patients specifically and what do they do. So now, if you look up mutations in this huge gene, in database of clinical variants, you will see that the number of these mutations is just enormous. If you dig a little more, you will find that a large proportion of these mutations has uncertain clinical significance. So we simply don't know what do they do. And this is something that we're trying to understand in our lab. So we are a biochemistry lab. So what we usually do, we purify proteins and then see what do they do in simple biochemical in vitro assays. In case of BRCA2, it's really hard to purify it, simply because it's a huge protein and it has multiple different domains as shown here schematically. And often when I present this work at the scientific meetings, people ask me how to purify the BRCA2. And I usually show them this picture. So you really have to start with a lot of human cells. And then after a few rounds of chromatography, you can end up with what is considered a pure BRCA2 protein, shown here on the right side of the gel. Now, sometimes people also ask me if it's expensive to purify BRCA2 protein. And then one day when I was bored, I just calculated the cost of all reagents that I use. And it turns out one needs about $20 to purify 1 microgram of this protein. Now, so those of you who do some investments might know that the gold is currently traded at around $50 per gram. So from that information, you can easily calculate that one can buy 1 kilogram of gold for just 2.5 milligrams of this BRCA2 protein. To me, this is just another example of the fact that biochemistry is simply priceless. Now, in the last few minutes, I'm going to tell you about one specific biochemical assay that we use to test protein and some mutants of this protein. So for the sake of time, I won't explain you all the details of this assay, but I would ask you to believe me that in this gel-based assay, the formation of this upper band, which is a product of the reaction, indicates that recombination happened. So in vitro, we can test whether DNA recombination happened. And so this, on the left side, you can see the two control reactions which we do. So first lane, when we add a RAD51 protein only, we see that recombination happens robustly. On the other hand, when we add the RPA protein, shown here on the second lane, this product formation and thus recombination is completely suppressed. So what it means that there is no way to switch from RPA on top here to the RAD51 on the bottom. However, when we add BRCA2 to the reaction, we can elevate this inhibition of RPA, of RAD51 by RPA by BRCA2 in a concentration-dependent manner. So this is a proof of principle. And just as one example, I'm going to show you one specific example when we compare the wild-type BRCA2 protein in this assay. It's shown here in the blue color to two different mutants of that protein, shown in green and red. And as you can see, the activities of these two different mutants are different from both each other and wild-type protein. So this is something that we're trying to achieve for many other clinically associated variants of BRCA2, and we also characterize them in some other different assays. So with that, I would like to thank my mentor, Steve Kolchukovsky, and all the members of the lab. Last but not least, all of you for attention, and thank you very much. I will be happy to take any questions.
Very interesting, and let's discuss now about maybe more about the function of the gene and the meaning of these mutations to clinical practice. Although I know that your main interest is molecular biology of this. But can you clarify us a little bit about what we know now about the function of BRCA2 in normal physiology? That's the other, and then the other. Please.
The major function of BRCA2 normally is to mediate this replacement of this one protein RPA for another RAD51 shown here. So this is defined as a mediator function of BRCA2 protein. That's what happens. So now, once you have a BRCA2 gene malfunctioned, this cannot happen. And because of this, this rather error-free molecular mechanism of homologous recombination is substituted in the cells with some other pathways, for example, non-homologous end joining, or some other ways to repair DNA through other mechanisms, which are generally much more error-proven. And because of that, cells accumulate mutations, and very often these mutations can lead to development of tumors. I also should point out that very interestingly and actually very beneficial to some of the patients with breast cancer and actually some other types of cancer. Once you have already tumor, which might have been formed by this mechanism of inactivation of BRCA2 gene or by some other means, but at the same time, if it possesses a somatic mutation of certain type in this gene, then it might be sensitive to specific drugs, and in this case, this is PARP inhibitors. So this inhibition actually works by the mechanism of synthetic lethality, and this is very interesting. It's like a dual-edged sword. Yeah, so that's what I can tell about the mechanisms.
Okay, thank you. Then I have received another question, which is related to the clinical significance of these mutations, and it is obvious that it has a very huge clinical importance because the BRCA2 mutations are so common in breast cancer patients. But is there already at this point, does it dictate the treatment of breast cancer? And maybe some other speakers could also answer this. For instance, Katri, who is oncologist, or Artem, how do you see?
I can tell from my that so far only accepted treatments I know of is PARP inhibitors, and to some extent it is determined whether to use them or not is whether the patients do have any problems with BRCA2 genes. But so many different mutations, they have no, we don't know about them at all. Thus we cannot say what's going to happen to this. We hope that at least in part this work which we're trying to do will be able to answer some of these questions. So that's what I can say from my molecular biology point of view.
Mm-hmm. Katri, do you want to comment this as a clinical oncologist?
Sure. So with the standard BRCA2 mutation, we use PARP inhibitors in ovarian cancer. That's standard treatment. But in breast cancer, it's not so well established yet. There are ongoing trials to see if the patients benefit from PARP inhibitors with BRCA2 mutations.
Thank you. And then we have the last question to Artem and also maybe the others, and that is that what is the future and what is the current situation concerning the screening, genetic screening of patients in order to identify those who are have a higher risk for breast cancer?
Yeah, so again, I can... No? Yeah, so I can comment, sure, yeah. There are probably people who know that better than me, because we mostly focus on fundamental science, but there are clear predictors for the cancer proneness. There's famous Ashkenazi Jew mutations, for example, which are the truncation of the protein, etc. So there are standard tests that can be offered to potential high-risk group people. I'm not so sure about that huge spectrum of other mutations, just because there are so many of them, and it's really hard to screen for something which you're not sure what to do. That's what I can comment about it. Yeah.
Katri, you want to comment this from the point of view of oncology?
So in Finland, we do that if the patient that presents with cancer, if she's young or if he's, well, we're talking about BRCA, if she's young, she automatically gets tested. Also, depending on the family history of a lot of more cases of cancer in close relatives, then automatically, yes, the patients get the test they want to. And all certain types of cancer, they are offered the test even if you're older. Like, for example, with triple negative breast cancer, they get the test if they want. But this applies to the well-characterized patients, for which we already know the clinical significance. So for the new one, this is data.
Very good. I don't see any further questions to Artem, so we thank you very much, and hope that can take a nap before the new working day, and we go on to have a presentation that is given by Katri Selander, who is an associate professor in the University Hospital in Oulu, University of Oulu, and Katri's topic is liquid biopsy cancer diagnosis and treatment. And please, Katri.
Okay. Unfortunately, I'm not associate professor. I'm only a docent. But anyway, can I get my first slide, please? To get into that position, I'll take any help. So I'm talking about liquid biopsies, which is an up-and-coming diagnosis to follow up cancer patients. Can I get the next slide, please? Okay. So liquid biopsies are basically analysis of any tumor-derived material that is circulating in the blood or in any other bodily fluid, such as spinal fluid or even urine or sputum, but mostly we're talking about blood. And when we talk about liquid biopsies, it means that the patient is, a blood sample is taken from the patient, and then we either get cell-free DNA that's floating around in the blood, or we're looking at circulating tumor cells. And remember that now I'm only talking about cancer. This is also used in fetal diagnostics, but my view is, of course, cancer. And my talk is focused on DNA, part of the liquid biopsies, because that is most commonly used. Next slide, please. Next slide, please. So these are the differences between the standard biopsy or liquid biopsy. So when a patient is diagnosed with cancer, for example, this patient in the image A, he or she would have a lung cancer, which is the big green circle in his right lung of the picture. So typically a needle biopsy is taken, and then the pathologist looks sample and says that yes, this is a non-small cell lung cancer or whatever lung cancer. We get the biopsy, we plan the treatment, and then the standard treatment occurs. When the cancer metastasizes, it typically changes so that there's further mutations and it keeps changing. And for example, in the liver, so the green ones are the original. Then in the picture, metastasis, the purple and orange ones are cancer cells or metastatic cells that have further mutated. And now if we take a like a standard needle biopsy, the needle may point or shoot at the purple cells or the orange cells or the green cells. So it only takes a biopsy or represents one small area of the cancer. So it doesn't necessarily give the treating clinician all the information that he or she needs to treat the patient. But don't go there yet. Can you go back to the previous slide? Okay, well, I was still talking about the previous slide. So in the liquid biopsy, you see that the needle goes into the circulation and the sample is more representative of the whole situation. You get samples of not only the initial cancer cells, but also the mutated ones. So now you can go to the next slide, please. Next slide. Okay. So what are the questions that the liquid biopsies could answer? We can ask that it could be used in screening, that is cancer present, and based on the answer from the liquid biopsy, it could give us hints that where is the cancer. We can also ask the cancer spread, has it metastasized to other locations? Using the liquid biopsy sample, we can answer what genetic changes does the tumor have? We can also use this liquid biopsy to answer that what treatment might work. We can ask, are the treatments working? And then we can ask that, is the patient cured? Is there any indication of unseen cancer left? So these are the questions that we answer with liquid biopsies. Next slide, please. So as you know, cellular DNA is typically in the nuclei, and in this image, nuclei, of course, are the blue circles, blue dots. Next slide. Next slide, please. But in the liquid biopsies, we are at cell-free DNA. So DNA that has escaped the cancer cells into the circulation. And the way the DNA can escape from the eye of cancer cells is through cell death. And cancer cells can die through several different mechanisms. For instance, necroptosis or apoptosis or necrosis. And upon cell death, small fragments of DNA from the cancer are released into circulation. Next slide, please. So if we compare cell-free DNA that comes from healthy cells or cancer cells, so there are certain differences. For the healthy cells, the cell death rate is very low and cell death is very rare. And once the cells die, the white cells clear extra DNA and very little DNA ends up in circulations. But in cancer, cell death is very frequent. The scavenger system is overloaded, meaning that the cancer cells, the white blood cells cannot clear enough of the DNA that gets released into the circulation. So it spills over to blood. and lots of cell-free DNA ends up in circulation. Next slide, please. So cell-free DNA typically is 150 to 200 base pairs long or short, and like I said in the beginning, detected in blood, urine, pleural fluid, CSF. Even in patients, there is some amount of cell-free DNA in the circulations, 1 to 5 nanograms per ml approximately. But in cancer patients, this concentration is much higher. It can be up to microgram amount per ml, especially in patients that have a high load of tumor. The half-life of this DNA is short, however. Slide. So once the DNA is isolated from the blood, the cell-free DNA is isolated from the blood, it can be analyzed with basically any DNA methods. It can be sequenced, it can be PCR'd, it can be analyzed for methylation, etc. Anything that you can do with DNA, you can basically almost cell-free DNA. But typically, we look at mutations in oncology. Next slide, please. And this is how we in oncology use the information. So once again, on your left hand, you see analysis that are done with circulating tumor cells. I'm not talking about those so much, but on the right-hand side with the purple images, you see like mutations that we can look up from the cell-free DNA. And for instance, we can look at epidermal growth factor receptor mutations, for example, in non-small cell lung cancer. We can look at KRAS mutations in colorectal cancers. We can look at PIK3CA mutations in breast cancer patients, and so forth. Next slide, please. So this is much more used in the United States as it's used in Europe, but this is coming to Europe already. We use these things to a certain extent also in Oulu, in my hospital. The indications for the use of this kind are increasing. The more we know, the better we can use these tests. The clinical feasibility of these tests is well established. We know that what we measure is correct, but the clinical utility still hasn't been shown for everything, meaning that does the patient benefit from this information? We of course all predict that it will be shown, but it hasn't, officially been proven everything. And in Finland, the way, for example, how I use these tests is that if I have a non-small cell lung cancer patient who does not benefit from the first EGFR receptor inhibitors anymore, I take a blood and ask, does the patient have T790M mutation in this cell-free DNA fraction? And if he has if he has this mutation in his blood, we know that it's derived from his cancer, and we know that he's going to benefit from osimertinib, and I know to switch him on this other second-line treatment. And we are starting to use also these liquid biopsies in breast cancer patients that have metastasized breast cancer, hormone receptor positive. They do not respond to CDK4/6 inhibitors anymore. This test and ask if they have PICK3A mutations in their cell-free DNA fraction, and if they do, I know that there's a good chance of them benefiting from drug alpelisib. Next slide, please. So that's all I had to say. Kiitos, thank you, spasiba. I'll be happy to take any questions.
Thank you, Katri. And there are actually a number of questions. And the first one is related to the sensitivity of these cell-free DNA measurements. Of course, some specific genes that you are after. So how do you see for instance, is there any possibility to use this type of approach, for instance, to follow how the treatment affected, has tumor disappeared, and so on? So do you have any idea of the sensitivity at the general level?
Yeah, well, because it's based on PCR, it's highly sensitive. Very few. What? So yes. So if it's positive, yes, it's highly sensitive. It's PCR. If the person was first positive, and then if he becomes negative, it's repeatedly negative, then it's likely that it's true. I can't give from the top of my head the specificity and sensitivity numbers, but it's well established. Nevertheless, if the patient has a lot of tumor and there's, for example, in this, and you get a negative response, that doesn't prove anything yet.
The second question is also related to the same topic, and it is so that is there at the moment, are there any studies ongoing, or are there expected to see one can use this liquid biopsy, cell-free or maybe also the containing cells, or for genetic screening possibilities to identify the cancer?
Go to clinicaltrials.com, type in a liquid biopsy, and you're going to come up with hundreds of clinical trials that are ongoing. This is a very hot topic, and this would be very highly helpful test because non-invasive, much easier, taking a lung biopsy or a liver biopsy.
Mm-hmm. Okay, very good. Well, I think that I don't see any new questions. Oh, there is one. It's related to that. Why the half-life of DNA, this DNA, is so short?
There's DNases in blood. They quickly chop it up, and if you have cancer that keeps, spilling DNA into the blood, you have a chance to capturing it. But in healthy conditions, the system clears it so fast. But DNA is the answer.
Okay. Very good. Okay, at this point, I thank you very much. And we move on. And our next speaker will be Dr. Yulia Kudryashova. She is a chief consultant endocrinologist in State Hospital number 28 in St. Petersburg. Her topic today is genetics and diabetes, clinician's perspective. Julia, please.
Hello. I would like to thank you for the opportunity to participate in this conference today. I'm an endocrinologist, so on daily basis I deal with the clinical application of genetics findings. So my presentation. So today I would like to discuss the landscape of type 1 diabetes. Next slide, please. According to the International Diabetes Federation, there were 463 billion people worldwide with diabetes in 219. Next slide, please. So this means that 1 in 11 adults suffer from diabetes, and this also means that there are going to be 700 billion people diagnosed with diabetes by the year 2045. And we need to remember that diabetes is one of the leading causes of death worldwide. Next slide, please. Diabetes is a group of metabolic diseases which is characterized by hyperglycemia resulting from defects in insulin secretion or action or both. The chronic hyperglycemia of diabetes is associated with long-term damage and dysfunction of different organs. Next slide, please. There are both symptoms of hyperglycemia that could be acute and chronic. Both those symptoms could lead to premature death. Next slide, please. There are several types of diabetes, and I would like to focus on type 1 diabetes. Next slide, please. Type 1 diabetes is an autoimmune disease which is characterized by autoimmune beta cell destruction, and it leads to absolute insulin deficiency. So it requires lifelong insulin therapy. Type 1 diabetes represents approximately 5 to 10% of all cases of diabetes. There is no significant gender before the age of 15, but after the age of 25, the male to female incidence ratio is approximately 1 to 5. So there is also a significant seasonal variation in the incidence of the disease. Lower rates are in warm summer months and higher rates are during the cold winter. Next, please. Next slide, please. The measurement of all antibodies in venous blood could reveal active disease years before clinical diagnosis. And this is very important because we don't have symptoms, we don't have clinical manifestations, but we know that we need to focus, we need to keep in touch with those patients. And the most important question for us as clinicians is how to find those people whom are we going to test. So here genetics findings play the most important role because we can stratify people and find who we need to test for antibodies. So next slide, please. I would like to show you this picture because the incidence of type 1 diabetes, you can find it all over the world, but there are certain countries where the incidence is higher, and the highest are in Finland and on Sardinia, and the lowest are in China. Next slide, please. There are several factors. Next one, please, next slide. There are several factors that trigger and react from the development of type 1 diabetes. So after the genetic screening, we could focus on those factors that could be modified. Nowadays, we have these beta cell stress hypothesis, which tell us that there are several factors which could drive pancreatic beta cell destruction alone or combined together, which include overweight during childhood, different deficiencies, stress levels, trauma. And I would like to pay your attention that vaccines, and it's very important, and also cow milk were excluded from possible triggers. Next slide, please. Next, thank you. Type 1 diabetes is largely heritable, has largely heritable component, and twin concordant rate is 70%, and also the risk is about 8%. Two DRQ haplotypes at IDD1 loci are by far the most important factor and the most important finding in heritable type 1 diabetes risk. And also the region, HLA region on chromosome 6q21 contributes approximately with 40% to type 1 diabetes incident. Next slide, please. There are several most important loci, which include IDDM1 that estimated nearly 40 to 50% risk of developing diabetes. The next one is IDDM2, and then it's PTPN22, which is responsible for less than 10% of cases. Next slide, please. There are two main approaches in dissecting type 1 diabetes genetic backgrounds. It's linkage analysis, which looks at the relation between the loci and the disease trait within family, and association analysis focuses on the relation between a specific allele and the disease trait within population. Next slide, please. Linkage analysis is used for the genetic mapping of Mendelian and complex traits within families aggregation. Here we use siblings, and those analyses are relatively rare because it's really hard to find large quantities, large amounts of twins with type 1 diabetes. But this analysis is really efficient in finding rare alleles with a large effective size. Next one, please. Here you can see you can see how we use those genetic findings. So we try to stratify, we try to find the risks, and then to apply those in our clinical practice. Next slide, please. Association analysis approach focuses on the relation between a specific allele and the disease trait within population. So we could use case control design, family-based design, genome-wide association studies, which are now the hot topic, and most findings are from genome-wide association studies, and we could use other types of genetic research. Next, please. Case control designs are the most common types. And here we used groups of individuals with the disease and without the disease. We also use genotype markers with SNP with the disease of interest in case control and family samples. And next please. Next slide, please. Okay. Family-based design, we use parents' offspring trace, and it is really a great approach when the age of disease is relatively low. And also it is used to rare phenotypes within the family. Next, please. Next slide. Thank you. Genome-wide association studies have already enabled us to identify more than 50 loci that contribute to the risk of developing type 1 diabetes. And also those studies, and I think that it's one of the most important from the clinical point of view, that those studies found some key elements of disease etiology of type 1 diabetes. Next one, please. So, next slide, please. So now we have this combination linkage studies have demonstrated that multifactorial disorders, including type 1 diabetes, cannot be explained by a limited number of rare variants with large effects, and association studies have shown that they cannot be explained by a limited number of common variants with moderate effects. So the most significant gap is currently in detective low-frequent variants with immediate effects. Okay, and the last one, please. Here we come to the clinical application of genetics findings. First, we have primary and second preventions before clinical intervention, and at the first step we need to stratify the genetic risks, and then there are secondary prevention measures, which are control of inflammation, introduction of immunological tolerance, and sometimes try to restrain the immune function. At least we have some clinical trials which are dedicated to this method. And the last one, when we initiate treatment, and genetic findings sometimes help us to find the best possible treatment. Thank you so much for your attention. Kiitos. Спасибо.
Thank you very much, Julia. And now I wonder if there are any questions. At the moment, I have not received them to my mail, but let me start by asking that how do you see, Julia, on the basis of current knowledge, that is it more like an infectious disease, or is it more like a genetic disease, this type 1 diabetes?
Thank you for your question. I have to deal with a lot of patients who are in distress because their relative have type 1 diabetes, and they come to me and they ask for guidance, for clearance. And what we tell them is, of course, there is a strong genetic evidence, especially when it comes to siblings, but also we need to focus on preventing the exposure to viruses, especially at young age, and especially when the woman is pregnant. So those are the easiest steps to stratify risks. And also it is very important to focus on eliminating all kinds of nutrient and vitamin deficiencies during the childhood. A lot of trials are there nowadays for people who have, ancestors or siblings with diabetes. So we try to have complex approach. So you could develop type 1 diabetes without inheritance, and also there is an opportunity not to develop type 1 diabetes at all if you have a sibling with diabetes. So it depends on luck, actually.
I know we have a very interesting vaccine clinical trial ongoing at the moment in Finland.
Yeah,
And
I know about it. Yeah.
how do you see the genetic screening at the moment and in the future? That is there a place for the more general screening concerning type 1 diabetes? I mean, among the risk groups.
In my opinion, genetic screening is really important because we can find better clinical approaches, especially when there is a vaccine on clinical trials. So I think that we mostly focus if the patient has family history of type 1 diabetes, or if the patient have suffered several viruses, including Zika virus, rabies virus. So we need to have some background because general screening, when we screen large amounts of people, no, there is no clinical evidence whatsoever worldwide. So we still focus on family history or viruses exposure.
I see. Are there questions from other speakers? I don't see any hands waving. So I thank you, Julia.
Thank you.
It was very interesting. And then we move on. And our last speaker is also Julia. And she is Julia Sopova. And her topic is, actually, Julia is the senior researcher from Vavilov Institute for General Genetics in St. Petersburg. And her topic today is genetic tests for antibiotic resistance. So we are now moving from group of diseases towards the bacteria mainly, I suppose. Julia, please.
Hi, my name is Yulia Sopova. I'm working at the Vavilov Institute of General Genetics, and this is part of our work together with Scientific Research Institute of Children's Infection here in St. Petersburg. I would like to thank you all for opportunity to participate in such interesting meeting. So I will tell you a little about how can we test for antibiotic resistance, not in usual way, but in a genetic way. First of all, what we know that bacterial resistance to antibiotics leads to very hard problem, because when you have antibiotic resistance, you cannot use antibiotics at all because bacteria are still living. And how happens this antibiotic resistance? First of all, the main cause is just the use of antibiotic itself. So when you use antibiotic kills all susceptible bacteria, and you have some bacteria that are drug resistant. Another opportunity to spread this antibiotic resistance is the transfer of resistance strain from one person to another, or even from non-human sources to human, and these non-human sources may even include food. So we can try to detect this resistance before we can manage the person who is ill, and in a clinical way, the standard testing are dilution methods, or agar disk diffusion testing, or gradient diffusion methods. But all these standard methods, they require days to complete it. So you have patient, you try to cure the illness, but still you need to wait about 1 day or even more days just to know what's the minimum inhibitory concentration, it's called MIC here, is, and whether you can use antibiotic or whether you can try another one. So it takes time, so we need to do it faster, not in days but in hours. And to our help is the simple method, it's just PCR. So you can have suspension of bacteria, you can perform total DNA extraction, yes, and then you have PCR, you amplify selected region, you have electrophoresis, and in the end, after several hours, it takes hours, about 7 or 10 or even 1 day it might take, you have the result. If you have this specific band, then you have a resistance to specific antibiotic, and this method is very simple, but it has some limitation. For example, you just need to know what gene you are looking for. And this method has some disadvantages. For example, you might have non-specific reaction here. And still, this method is used, and it's used for example, for detection some antibiotic-resistant genes in enterobacteria, for example, resistance to cephalosporins, carbapenems, and so on. But another way to do it faster, so we need to do it faster, is single real-time PCR or multiplex real-time PCR. What's the difference? In endpoint, you need to wait till the end of electrophoresis. Here, you just can look what is happening in real time, and you have this parameter, it's called CQ. It's the start of exponential phase of this line of amplification. And if you have specific probes, for example, you have specific primer number 1, specific primer number 2, you have specific Tachman probes, you can measure fluorescence. So this method is quite specific. It's widely used. It's used for detection of main antibiotic resistance in enterobacteria. Very important that it's used in S. aureus, methicillin resistance, cause of many dangerous infections of human. And of course, you can use it just for direct detection of bacterial DNA from clinical samples. For example, for chlamydia, mycoplasma, or Neisseria, this complex of infections. And this method is widely recommended by different guidelines. For example, in Russia, you can buy commercial kits to do such analysis. But still, we have to do it faster. So hours, we need to do it in minutes. We have some new methods, for example, this integrated PCR and high-resolution MELT. Again, we have PCR, but better to have here on this line quantitative PCR, because here you grow the bacteria with the addition of antibiotics or without drug. Then you have the difference here, CQ delta, resistant or non-resistant, and you can identify the bacterial species by melt curve. So you can just compare and have the difference. But still we need to go faster. To go faster, we have a very special, very new method. It's called LAMP, or loop-mediated isothermal amplification. Now we have 4 specific primers to our target, and this method is more sensitive than all conventional PCR methods. You can use it for detection of meningococcus or even for E. coli. But again, we have some powers. And the fastest known method now, it was published in Science Translational Medicine, it's digital LAMP. So again, you have LAMP, you have digital, you have just sample, for example, this urine published, incubation without or with antibiotics. Then you have selective AST marker in digital real time. So you have more than 1,000 wells just to analyze, not one well for one sample, but about 1,000. And then you have, again, compared ratios. Either it's resistant or it's successful. It was further tested in this year. It was the publication about Neisseria gonorrhea. And still, when we have all these tests, we need to know what gene conferring the resistance we are looking for. To know this, we need to perform whole genome sequencing. Next generation sequencing gives us very important information about whether this resistance did come from a new gene, or is it a mutation in gene that is already known. So we have clinical sample. We can either cultivate it on selective media and have pure culture, or extract DNA from sample directly, then sequencing, then we can assemble the genome, and after it, what we are doing? We have databases. We can search for genes that are connected to virulence, and they are different. We can search for different genes, and so here's one publication that was made about methane-resistant Staphylococcus by my colleagues. When we have this genomic data, we can either know which origin of resistant gene we have, or we can even search for new possible resistant genes. So I should thank my colleagues from Scientific Research Institute of Children Infection, Vladimir Gostyev, Vladimir Ageyevets, Irina Lazareva, and of course the head of the department, Sergei Sidorenko. And thank you very much for your attention. Kalerva?
Yeah, thank you very much, Julia. Can you hear me?
Yeah, but I'm trying to switch to see you, because just a moment. Yeah, here I am.
Okay, very good. And how it is at the moment? Is the genetic methods overwalking the classical traditional microbiology in bacterial laboratories today?
No, they are not overworking. They are just working together. You have classical clinicians. They are usually very skeptic when you have such molecular, something AT, GC. It's very hard. It may be very hard for them to rely on such data. When they have a minimum inhibitory concentration, it's simpler for them to rely on it. But still, I can say that these classical and new genetic methods, they must work together. So not only genetic methods, but of course you better to do them both. So I can see.
I see. Are there questions from the other speakers or from the audience? I don't. Julia, is there something burning issues you want to add at this point? We still have a couple of minutes time.
No, I think. Hope that it was interesting for people who are in medical genetics to hear something from the world of bacteria.
Yeah, that it was very interesting. And this genetic testing will be the first laboratory in this Russian-Finnish life science park to function. So this is why we have chosen the topic to focus on several different aspects of genetic testing in medicine. So, and I think that our meeting has very clearly shown that genetic testing is becoming a very powerful tool in many areas of medicine, coming from the let's say systemic diseases and infectious disease, and of course especially in cancer biology. And it is I think already very clear that there are, it will have a more and more bigger role in the future in several diseases, both in prevention, in diagnosis, and of course also to follow up how the disease is progressing, and also in determining the best possible treatment. But now I don't see any further questions, and we have heard all our presentations. I wonder if Yelena Petrova, do you want to say something at this point?
I thank everyone for participating in the conference. Thank you, dear colleagues, in our conference. Very well.
Okay, thank you, Yelena. And as a moderator, then I also would like to thank all the speakers. They were excellent presentations. It's very difficult in this type of seminar when you don't see the audience and you don't see the responses from the people immediately to follow what's going on. But as far as I have the feeling now, I think we make this a very nice meeting, having different topics, and I hope that there will be further collaboration between different institutions on the basis of this. We will definitely continue this type of small conferences. Let's hope that at some point we can do it also in real life and in I mean face to face. But meanwhile, I think that this is also a very nice way to distribute knowledge across the borders using this type of video conferences. So thank you very much for all speakers and all participants, and have a nice summer and safe summer. Thank you.
Thank you. Thank you so much.
Thank you.